Hi everyone, welcome to BIS103 lecture 11 again. This is actually video part 2 for this lecture. So if you haven't watched the other video that focused on the mechanism and the nature of the electron carriers that are really critical for electron transport chain, I encourage you to watch this video first before you are focusing on this one, because it will be very difficult to understand the energetics, of these reactions that we want to discuss here without this foundational knowledge of the processes of the ETC. So for this video, I really want to look at two things. One is to understand the energetics of why transferring electrons across the electron transfer chain can be used to facilitate the transfer of protons across the membrane and ultimately ATP synthesis. In addition, I want to look at least a few examples of experimental approaches that people have been used in the past and that are, at least in part, currently still being used to study and understand the processes of the ETC. So learning goals, just again here, what we want to achieve here is that we want to be able to describe different experiments that can be used to study the redox and oxidation reactions in the ETC and the electron transfer processes. And we want to understand and actually apply how we can calculate the bioenergetics in this system for electron and proton transfer in the ETC and how this relates to ATP yield. So let's start with some of the experimental approaches that people have used. There are many more. Just sort of in the interest of time and our scope here, we'll only focus on a few. One really common one that is still being used, not just for the ETC, but many other biochemical work, is that you can isolate the enzymes that are important here and use them for biochemical characterization to understand the function of these enzymes. That's one, that's an in vitro technique. If you want to go in vivo, so if you actually want to understand these processes within an organism or at least a cell, you can use inhibitors. Right? Inhibitors, chemical compounds to inhibit pathways. You today could also use genetic mutants, but in this case, it's very common to use inhibitors that block individual complexes of the ETC so you can understand how they work in concert with each other. So how does this work? Just here to recap some of the material we have covered in part 1 of our video on the lecture on the ETC. Right here you have your mitochondrial inner membrane here. This would be the inner membrane space, so the space between the inner membrane and the outer membrane of the mitochondrium. And then on the inside here, they have the mitochondrial matrix, so the inside of our mitochondrium. And then in the inner membrane, we have our 4 complexes bound to the membrane, our complex 1 here, complex 2, 3, and 4. And then we have our CoQ, small molecule as an electron carrier, another small protein here, cytochrome C, that is partly mobile within this membrane and can transfer electrons from complexes 3 to 4. So just in a nutshell, right, what these do is they're taking on the electrons from our electron carriers, NADH and FADH2, transferring them through the complexes, and then ultimately complex 4 would transfer the electrons over to oxygen. We are generating water. Right? And in this process, we're using this energy to push protons across the membrane in 3 of these complexes, and the protons then later flowing back can be used in the ATP synthase to generate ATP. So this all looks very tidy, but people have worked hard over decades to understand how these complexes work, to identify them in the first place, and then to understand what is a series of reactions that is actually happening here. And so using inhibitors can be really helpful in this case. Right? So let's look at a few examples and see how we can do this. So say you would use inhibitors of complex 1, right? So there are some examples here, barbiturates, which are sedative drugs, for example, or rotenone, which actually is a pesticide. They can be used, and so these are now small molecules that inhibit complex 1. So if you do this, what happens, right? What happens now is that you cannot actually transfer electrons from NAD to NADH anymore. So two things that would be happening here is you're obviously losing out on the power of electron transfer because you're losing out of all the NADH, and NADH would actually accumulate. Right? If it accumulates, it only gives you an idea that if you're blocking this complex, its function obviously is to oxidize NADH and to transfer those electrons. So from blocking it, you already can make inferences toward its function. Right? You would also observe in this case that FADH2 is completely unchanged. Right? It does not go through complex 1, so your second observation would be the function of complex 1 is not to transfer electrons from FADH2. So these two observations together, by simply using an inhibitor, gives you an idea of what these enzyme complexes are actually doing, what their function is in the ETC. Another really important point here is that we can highlight by this experiment the key importance of CoQ here, and I alluded to this in the other video, as a key metabolite that allows the branching of the ETC. Right? CoQ, because of its mobility within the membrane, it can move between these complexes, and it will take on electrons from complex 1 and complex 2, as well as a number of other dehydrogenases that we talked about in the other video. It will take all these electrons and shuffle them through to complex 3. This branching now becomes a fail-safe mechanism, right? If you would assume, right, you have overdosed on barbiturates, on these sedatives, your complex 1 is blocked, right? If you would only go from complex 1 to 2 to 3 to 4, you would not be able to do ETC anymore. This would be fatal, right? You cannot actually make ATP anymore, and you need ATP to survive. But because of the branching here and of the role of CoQ, it could still take on electrons from FADH2, so you could bypass the blocked complex 1. You can still run the ETC using the electrons from FADH2 because CoQ is grabbing them from different positions here. Right? Yes, of course, the ETC would be much less efficient here, but you still can make ATP, so you might actually well survive. So this branching of the ETC is actually absolutely critical for living systems. What about some other examples? What about the other complexes? Right? We can use the same kind of strategy. Say you're using something like antimycin A. It's used as a fish poison, but what it actually does mechanistically is that it inhibits complex 3 now. Right? And so in this case, right, this is a complex that is taking all the electrons over from CoQ, right? Regardless of where CoQ gets its electrons from, whether it's complex 1 or complex 2, it gives it to complex 3. So if you block this complex here, there is no ETC happening. That would be fatal, right? But because of this, right, now you would observe in your test system that you're accumulating both NADH and FADH2, and you're also accumulating reduced CoQ. So all of this will tell you where the ETC complex 3 is functioning. Another example here is cyanide. You may have heard this in all of the Bond movies, right, where people use cyanide. This again, actually the way cyanide works is that it blocks complex 4. So it would function in a very similar way as blocking complex 3 here. It would be fatal. You cannot do the ETC anymore. You can use it, so like the combination of all these inhibitors together, you can understand which of our carriers are accumulating in their reduced form, which of our complexes are accumulating in their reduced form, and looking at these differences using these inhibitors, you can understand in which series are these complexes actually operating. So it's a very simple but efficient system of understanding multi-complex pathways. Right. Another thing you can do also would be you can culture mitochondria and look at this in an in vivo system using live mitochondria that you have isolated from yeast systems or other animal systems, for example. So assume now an experimental setup where you would isolate these mitochondria and you would remove all the oxygen in the solution with the mitochondria. So you can achieve this, for example, by bubbling in nitrogen. It will get rid of almost all the oxygen. Once you've done that, next you're adding pyruvate. You can buy that. It's not actually too expensive. what it does is right now your PDH will be active and will convert all the pyruvate into acetyl-CoA, will release NADH, and in the process, what it will actually be doing is it will use up everything, right? Now you have NADH in the system. That means your electron transfer will be running in your live isolated mitochondria, but it will only do so until oxygen is completely gone. So all the oxygen you haven't removed by bubbling in nitrogen will be used up by feeding in NADH and having the ETC run in your isolated mitochondria. What you're achieving now is a situation in these mitochondria where there's no oxygen. So your complex 4 cannot actually give its electrons away to oxygen. That also means everybody else gets to a hold, right? So actually, because of this, you have run the system and all your complexes at this moment are all reduced. Okay. And so when you come to this, you hit the start button. What you actually do is you add oxygen back in. So you let the whole system run again. And you can observe now what is actually happening. So I try to illustrate it in this diagram here, where you add oxygen here at time 0. And what you're now observing over time here is the percent of your oxidized carrier. Okay. And so essentially, now that you're pumping in oxygen again and you start running the ETC through all the complexes again, you would see curves of increasing amounts of now the reoxidized carrier. And essentially, the more steep this curve is, the faster this carrier will have been oxidized in the system. And the speed of oxidation and the relative amount of oxidized carrier over time, you can actually use again to understand the series of reactions. So in our case here, if you have a graph like this, you could deduce that the moment you put oxygen in, right, the complex that is actually acting the closest to oxygen, so next to oxygen, giving its electrons directly to oxygen, would be complex X, because it is oxidized the fastest, right? So it's the first to be oxidized. The amount of oxidized carrier happens the fastest. It has the steepest curve. Then the middle one here would be your next one acting downstream. And the lowest one, so the one that is slowest to oxidize, is your complex Z here. So it has to wait until complex X and Y are oxidized before it can be oxidized, because it would now give its electrons to these other complexes. That cannot happen until these are oxidized. So you can imagine it basically as a traffic lineup when the light is red. So the moment this goes to green, this guy can go, but these guys can't go. They cannot get oxidized until this guy has left. So if you sort of want to have sort of a simple example on how you can imagine this, this would be a good one. Okay. And again, these experiments can be used to understand how these complexes work in concert with each other and what the series of oxidation reduction reaction actually is in the ETC. And then last but not least, we can also use energy again to do this, right? What we can actually do is we can calculate the energetics of these reactions, much like we have done for our Gibbs free energy when we talked about pathway directionality. Right? We can do this too specifically for these oxidation reduction reactions. And here what this is called actually is a standard reduction potential, or SRP, which we give in delta E0 prime. Okay. So a similar idea, we're looking for standardized conditions that allows us to understand, are these redox reactions actually happening spontaneously? Are they exergonic? Okay. And this will give us, again, an idea on how we can predict which carrier acts in which series and how are they all working in concert with each other, similar to what we have done when we talked about multi-enzyme pathways. So how does this work? You can actually measure this, right? You can measure the SRP and can use it to calculate these values. And we're building it on the idea that the electrons will always flow from a carrier with a low SRP to a carrier with a high SRP. So if we want to use the SRP to predict which of the carrier is reacting after which, we are following this assumption of electrons flowing from low SRP to high SRP carriers. Okay. So it gives you an idea then of the tendency of the carriers to donate or accept electrons, and that we can actually measure. And so the way we do this here, I'm just using an image from your book here. You basically have two solutions here in a beaker. One is your reference solution here that you set artificially at 0. That's your reference. Usually these are protons and hydrogen mixtures. And on your other one here, you have now the electron carrier and an equal mix of oxidized and reduced form of whatever carrier you want to test. In our case here, let's just assume we're using NAD+ and NADH. We're standardizing it again at 1 molar concentration, constant pressure, and so forth, as we had done for our Gibbs free energy, our delta G0 prime. Now what we can do now is we can connect these beakers and the solutions with a salt bridge, so we can allow electrons to flow, and we can actually add a voltmeter to measure this. So we can now have different scenarios, right? We can have electrons flow either from the reference to the sample or vice versa. And so these values we can then use to calculate the SRP. And so there are two possibilities now, right? There would be one possibility where the electrons flow from the reference to the sample. Right? In this case, we would call it a positive delta E0 prime, which means that the carrier you have in your solution here would be a stronger oxidizing agent. So it's oxidizing other molecules, meaning it's a stronger electron acceptor. Okay. If the opposite happens, if your electrons actually flow from the sample to the reference, the opposite is true. Our delta E0 prime is negative. And so your carrier then is actually a stronger reducing agent and a stronger electron donor. And so this actually has been done. These values have been calculated for all the carriers that are involved in the electron transfer chain. So how can we use these values now to actually understand the bioenergetics of these reactions? So let's walk just through one example together here. How can we calculate the SRP and how can we correlate it with our Gibbs free energy? Because that's in the end what we want to know, right? Is, are these reactions exergonic in terms of their free energy? So as an example, what I would like to use is sort of the overall reaction of oxidizing our NADH cofactor that is coming into the ETC all the way down to the final electron acceptor in the ETC, oxygen, right? I'm using a half of oxygen here. I've realized that's biochemically not really accurate, but for calculating the stoichiometry of these bioenergetic reactions, we're using this half oxygen. So we're looking at the entire ETC now when NADH comes in. So our reaction is, right, reduced NADH reacts with oxygen. It will be oxidized. Oxygen will be reduced to water. Okay. Much like what we had done before for Gibbs free energy, we can break this down into the half reactions. In this case, it's called half cells. It's not so important, but just to be precise here. And so the half cells now are, right, one, the oxidation of NADH to NAD+, we're releasing 2 electrons and 2 protons. And the other half reaction is our reduction of the oxygen using 2 electrons and 2 protons to generate water. Okay. Now I had said, right, all these values have been measured, and I actually show you a table here. I apologize, the quality isn't the best, but just to highlight them for you, these are all these SRP values for the carriers and the half reactions that have been measured. Okay. Keep in mind, right, these are always in the direction of reduction, right? So you have to look at your half reaction, whether it's an oxidation or reduction, and look at these values, whether you have to turn them around, okay? And we'll use this here actually. So if we look at our 2 half reactions, right, the first one here, our oxidation of NADH, if you sort of go through your list here, your NADH is listed right down here, and it has an SRP of minus 0.32. But right, shown here in the direction of reduction, right? So you have to turn this around, so it's actually plus 0.32. Our second one now is oxygen, and in this case, it is a reduction. It's right here on top, right? In the direction of reduction already, we have an SRP of 0.816. And so those values we can now use. And much like our delta G0 primes, they are additive if you're adding up the two half cells. And that's exactly what I did here. So you have your two values here, right? Keep in mind, this is plus 0.32 because it is shown in the direction of oxidation. You can add them up and you have your SRP for the entire reaction. That's just a voltage measure. That doesn't really tell us much about Gibbs free energy and about spontaneous reactions. So we want to correlate this with our Gibbs free energy. And there's another formula that we can use that I will give you. You do not have to remember this, but we can actually correlate our delta G0 prime with our delta E0 prime in this formula here, where we're actually using minus n, which is simply the number of electrons that have been transported in this reaction. In our case, that would be 2 here, and our Faraday constant. So in essence, just the energy that is bound in 1 mole of electrons, if you will, just very, very basically. So if we plug our values now into this formula, what we get is actually a Gibbs free energy under standard conditions of minus 220 kilojoule per mole. That looks pretty good, right? It is negative, and negative Gibbs free energy means it's fluxing spontaneously left to right. It's an hexagonic reaction, so that's great. But what we really want to know is this enough to bring about the formation of ATP? So we have to remember from our earlier lectures how much we need to make ATP. And so I'll just pop it up here. And we had calculated initially that the phosphorylation of ATP, which is what we will be doing next lecture in our ATP synthase lecture, to ATP requires a Gibbs free energy of about 30.5 kilojoule per mole. So if you compare the two, this looks really good, right? So we are releasing lots of energy in the transfer of these electrons that we can now use for ATP synthesis. It's obviously energetically very well workable. So what I would encourage you to do at home as homework is to walk through all the individual half reactions of the individual complexes, just to sort of practice applying using these SRP values that I've given you here in this table, and using this formula that I have given to calculate the delta E0 primes and the delta G0 primes for the individual complexes. And sort of as guidance, I just have them listed here in the next 2 slides. I would recommend you do it by yourself first, just alone, and then see if you come to the same conclusions as I did for these different reactions that you see here, right? So there are 2 slides here that you can use, and we'll touch base on these actually in our next lecture, in lecture 12, on ATP production.